Our topic is electronic absorption spectra. The electronic absorption spectra refer to a range of wavelength or frequency of light absorbed by a substance, typically a molecule, as this transition between different electronic energy like S band, P band, and T band. When a molecule absorbs energy from light, electron are excited from lower energy to the higher energy state. Like if it is in the S state, it will excited and get into the P state, and if it is in P state, it get excited and reach to the D shell. This absorption occur at specific wavelength which depend on the molecular structure and electronic configuration. Key aspects like wavelength. The wavelength of light absorbed corresponds to the light difference energy difference between the electronic state. Like if there is a different wavelength, then it will excite a molecule electron to a D shell or maybe P shell. In the molecular transition, like in the DD transition, a electron will jump from lower D shell to a higher D shell, or from a lower pi shell to a higher pi shell, depending upon the nature of the molecule. What are the special features? The special features are absorbance, a measure of how much a light is absorbed at a particular wavelength. And second one is peaks. The absorption spectra usually displayed peaks at wavelength when electronic transition occur. UV vis range. Electronic absorption typically occurs in a UV and visible region of EM spectrum. Like if its wavelength is more than 800 Newton meter, then it will not emit any kind of electronic absorption, and if it is lower than 200 nanometer, then it is also do not excite any electron. Applications. The applications is like chemical analysis. Electronic absorption spectra help in identify compounds, analyzing the concentration and study of the molecular structures. And in the material science, they are used to study the semiconductors that are used in the electronics, and in the biochemistry, the absorption spectra is used to study proteins, nucleic acid, and pigments like chlorophyll. Luminescence. What is luminescence? Luminescence is a property of an object to reflect light. Like if we put a light on it, it will get illuminated. That is the property of luminescence. And we used it in a fluorescence imaging. The fluorescence dye and probes. The fluorescent molecules are used to label specific cells, tissue, or biomolecules. When excited by the light, they emit fluorescence, allowing researchers and clinicals to visualize cellular structures or biological process in real time. Like if we use any dye and then we give it the electronic torch or other different wavelength, then the specimen will be excited and will emit the light, which will help us to see and study the process that is happening under the microscope or under a specific thing. Fluorescence microscopy, a very powerful technique for observing the cellular components and molecular interactions with high specificity and sensitivity. Like we have a cell of a bacteria and we put it in the microscope and we put the fluorescence dye in it, and then we put the light on it, then it will illuminated and we can study it very well. Bioluminescence imaging. In vivo imaging. What is vivo? In vivo, when we take the sample in a live condition, that is in vivo. That is, it is used to study live organisms or living organisms, or to study the biological process like tumor growth and gene expression and other. This allows the real-time tracking of diseases progression. Third is chemiluminescence assay. Its diagnosis, like in the diagnostic what we used in chemical luminescence. Chemical luminescence is often employed in immunoassay like ELISA. In the ELISA, what we see? We will see the antigens and antibodies and pathogens in the blood or tissue sample or other sample that we use in the microbiology. What we can do, then we can diagnose what is the disease that is producing the antibody and antigen. Then the fourth one is molecular diagnosis, like the DNA and RNA analysis. Luminescence markers are often used in molecular diagnosis tools like PCR test, polymerase chain reaction, and DNA sequencing to detect genetic mutation. If we use the bioluminescence technique and if there is a problem in the DNA sequence, then we can see it very clearly and more clearly. Optical activity. What is optical activity? Optical activity refers to ability of certain substance to rotate the plane polarized light as it passes through them. This phenomena is exhibited by optically active substances which have a chiral molecule. What is a chiral molecule? It is a molecule which have a different functional group attached to it. Like if it is a C between it and it have 4 vacancy, then all on the 4 vacancy there are different molecules, like one have H molecule, one have CH3 molecule, one have OH, one have NH3. Then it is called a chiral molecule or chiral carbon. In what is the application in medical physics? Polarimetry for biochemical analysis, like glucose monitoring. Optical activity is used in polarimetry to measure the concentration of glucose in the biological fluid. Like we know that glucose is a have a optical activity, and when we pass the light through it, plane polarized light, then it will rotate the plane polarized light, and we can judge the amount of the glucose in that sample by seeing the rotation and frequency and the amount of rotation that is possessed by the glucose. More is the rotation power, then more is the glucose. Protein and enzyme analysis. Many protein and enzymes are optically active. Measuring their optical activity help in analyzing the protein structure, optic activity and concentration. Pharmaceuticals, drug enantiomers. Drug enantiomer is crucial for differentiating between different enantiomers of drug. Often one enantiomer have a desired therapeutic effect while the other may be a less effective or cause side effect. What is enantiomer? Enantiomers are the two chemicals that have the same chemical formula but different structures, and that is the enantiomers. And we see in the drugs that there are some drugs that have enantiomers and they have different function or may have different activity or may have a side effect. That is very important in the optical. So we have to we have to see which of the enantiomer we are giving. If it having a different optical activity, we can judge it by the different optical activity. And chiral drug analysis. Many drugs are chiral, and optical activity is used to monitor their production and ensure purity as different enantiomer can have vastly different structures with the biological system. There are some drugs that are chiral in nature, and if they do not rotate the plane polarized light, then we can see that they are not the product that we want. Chirality in biomolecular interaction, protein folding and misfolding. Optical activity can provide insight into protein folding as the tertiary and quaternary structure of protein influence how they rotate the plane polarized light.